Hello, everyone. It's really great to see you all here joining this workshop and the live session. So this workshop itself is a unique opportunity. And one of the reason is that because we in this workshop will cover the introduction to bioinformatics curriculum. And that is quite a comprehensive curriculum that would involve several things. So understanding how it is going to work is pretty important and very critical to this training. So we will dedicate a certain amount of time making sure that you understand the way this entire training is structured, including those who have recently joined. I see Nivedita here. She recently joined our research fellowship program for 6 months. Hello, Nivedita. Nice to meet you. And I also see a lot of other participants from our research fellowship program, like Dr. Pradeep is here, Dr. Priya is here, and it's really great to see you all. So we, in this workshop, will be covering the overview of bioinformatics, big data bioinformatics, and we will also talk about the application, and we will discuss how cheminformatics is also applied as a strategy to many different research problems. So this is going to be quite comprehensive, and as you have seen that we are covering a lot of heavy topics. So obviously we will not be able to go in depth of all those heavy topics, but we have assured that we have all the we make sure that you are getting to learn about all of that through different resources that we have put together for yourself. And that is something that we are going to discuss in this session, as well as throughout the workshop. We will be discussing how we will be easy to it how easy it is to access and do all of this, which is quite challenging at the beginning of when we all started with bioinformatics. So it's really nice again to meet you all, and I'm really excited that you have been able to join this session and join this workshop, and are eager to learn and apply some of this technology into your education or into your own research interest. So the first thing I would want to tell you all is that the OmicsLogic training has certain specific goals for each of you here who are joining. So we have students from high school. We have a lot of students doing research in high school for their student projects, and for their for to compete in different various scientific fairs and all of that. So these students are going through the same OmicsLogic training. So one of the reasons that is being possible is because of preparation of different kind of materials for different people from different backgrounds, students from different background. So students who are joining from high school, they can expect to be a part of this international bioinformatics research community, which actually helps all of us to be connected and help each other, right? And then we have a pool of experts who are experts in biomedical research, data scientists, biologists, and an opportunity to interact with them and discuss about your individual research questions, which is aligned and related to that, as discussed with by Sonalika previously, that our research fellowship program is just built for that. And then developing an appreciation of data in life sciences, that's kind of very critical to be able to move forward in this data driven era, where we are understanding that what data can actually do. Because something that we usually don't see in front of our eyes as a physical entity, but a data is present in the hard drives that stores this information and various type of other devices. So the actual growth and the effect of the data is kind of difficult to understand otherwise. But when you go through what it means and what it holds and how it can be useful and what it has been used for and what are the use cases, you will definitely understand that why we are appreciating that more and more now. Again, like this all leads to participation. One of the reasons you have joined here is that because you want to learn this, and also you want to showcase that you have been able to apply this to some of the things that are important topics for you. So that kind of exposure is required, and that kind of exposure can be gained through such training. And when I talk about students who are joining us from their undergraduate studies and from their master's studies, it has been really amazing because at the time when the OmicsLogic platform and bioinformatics was not so much popular, at the time when we were doing our undergraduates and masters, and that's what I meant. This kind of training was really difficult because the several things it requires. One, you can do a real research that is kind of really difficult for anyone who is doing an undergraduate study or a postgraduate study to go for such opportunity unless you get to a good lab, unless you've been getting an opportunity to work in an industry as an intern, and all of that are required for us to gain such experience at an early stage, at an undergraduate stage, at a master stage. Many of us who have done PhD could imagine that what could it be if we were doing and if we were knowing all of this way before. So it actually helps us design better career for us, choose better career for us, or think that what direction that I could contribute for in this world of science. Now the next level, there are few levels that I'm describing here, that level advances as our learning advances, and that level advances as we grow from one stage to another. So one of the things that we would want to expect many of you to accomplish is to be able to apply complex analytical tools and do modeling across multiple data types. And why modeling is required is kind of making sense, because when you have a lot of information, when you have a lot of data, when you have a lot of patterns, so one of the things that biologists or bioinformaticians would want to do is to develop prediction models. Because when you are going into the world of unknowns, you would want to have an idea that how can you understand from the unknowns, and that could be done by developing predictive analysis, by working on models, by able to do predictions in simple words that we do all the time for all of the life purposes. Similarly, when we do this, there's a need for validation. There's a need for validation from the community. There's a need for the validation from scientists, and also to be able to replicate something so that you are for sure that this is happening the way it is happening, that the way that you are proposing. So what we are going to learn today is also helpful in doing reproducible research, which is one of the key agendas in for doing research in today's world. Another thing is that how do we do all of this? Where do we get data from? And why we are talking about all of this, which would only make sense if you have the access to such data. Now, let's learn about all of that in a stepwise manner, and also let's learn about how this program and how this entire 1 month experience is structured. Because this is a three-day workshop, and we have had tremendous information that we got from yesterday's sessions, and those who were not able to join obviously can join. And we learned about NGS technology, one of the key technologies that is driving the advancements in healthcare research. How this is useful, we learned about the NGS basics, that how data is generated. Then we learned about what are the different clinical application of that sort of data, and then we learned about the advancements such as single cell genomics. So from there, we would want to transverse to why we are doing this again. So one of the reasons, obviously, that we see as a company is a huge gap. And that gap, I think, could have been observed by many of you as well. So this gap is about the understanding of data science and biology. So many of us who are really comfortable learning about different aspects and complexities in biology are not so much comfortable when it comes to programming, when it comes to applying data science or do advanced data analytics. One of the reasons is that there is a gap because for you to learn this multidisciplinary field and excel in this, there's a lot of requirement for you to learn about various things that would require a lot of hard work, a lot of time, maybe PhD. So how do we address that gap? We address that gap by finding the ways that we can incorporate resources and materials along with our mentor guided programs, along with our T-Bioinfo platform, which is an AI guided platform that we will explore throughout this workshop, is something that we've been able to achieve through OmicsLogic Bioinformatics education. I'm really excited that this is working for a lot of us, and we were able to understand that how biomedical data or how biological data could really change the way we do research. Now, there are few objectives that we will be following. The goal of this training is to enable independent research. So the ultimate goal of this training is that any all of you here should be able to conduct your independent research, and that is quite a difficult thing to achieve because scientists all around the world has the same objective. And at a student level, when you're doing your masters, when you're doing your PhD, when you're doing your bachelor's, it is kind of challenging to be able to do this research along with even your work. So if you're a doctor and you want to do research because you're generating so much of data, and you can see that you can see the patients, that how it is influencing, how what kind of drug that you're using, what is the effect, and your input is really really important, and that can change a lot of things for goodwill. So one of the reasons is that we do that. Along with that, I already told you that this community is something that you should be able to leverage. And now this community involves a lot of collaborators around the globe, which means that we have scientists from labs, from cutting edge labs in the US, in Europe, and many labs in India. So you can think about collaboration and utilizing this community, and then able to apply that into a real research work. Now, simplifying it is one of the key reasons that we are doing this session, the first session. So let's simplify all of it and what is going to be. So you are going to use some of the resources. These resources are online resources, so you'll be able to do everything online. And if you have a decent internet connection and a laptop, you'll be able to do all of the things that we are going to cover in today's session, including high throughput analysis, including molecular modeling, and all of that. So one of the first thing that you did, and I know that all of you have been added here, is on Learn OmicsLogic. Then in Learn OmicsLogic, what we are going to do is that it has to be very clear to all of you that Learn OmicsLogic is not just about online courses. It is also about to get hands-on experience. So hands-on experience can be done through data sets. When you are being able to apply these data sets into practice, and that's why this is called OmicsLogic, because you are understanding logic from a standpoint of data. Now, we will be using curated data set for practice in this session. And before understanding that how it works, it is really important because you will have access to the Learn OmicsLogic for 1 month, and then there is a subscription that you can take for $10 a month that would enable you to access all the resources that we have put together on OmicsLogic Learn. Now, how this you know uh, courses are structured is kind of important to understand, because unless you do that, you will not be able to make the most out of the courses. and the materials that has been shared with you, which is a key part of this entire training. We believe that that is going to help you a lot. So, Monica, I'm not audible to you. Am I audible now? Am I audible to everyone else?
Yes, Dr. Mohit, you're audible to us.
Thank you.
Maybe Monica, you need to rejoin. Oh, so yeah, that would work for you.
All right, Monica. So, going back to the structure of the course. So, when you start with the course, you will find that there are terminologies and then biological context. Then we are talking
Sir,
about
we
data
are not
form.
able to hear you, sir.
Okay.
Dr. Mohit, you are audible to us. I think Monica can rejoin.
Monica, can you try to rejoin again? She's not been able to listen. Maybe you can ping her and help her out, Sonalika. Okay, I mean, let's try doing that, and we are recording this session, so that is also there. So, as I was telling you, that when you start, you go through the terminologies, because for anyone who's starting from basics, and for example, when you are doing gene expression analysis and you don't know what splice variants are, and many of those top biological keywords, many of these computational keywords, so it's important to just understand their meaning so that you can go forward to the context, and then understanding why we are doing this for biology, and then coming to the data formats. Now, data formats are those are important to learn, because it is like, converting your biological information into a file format which is readable by computer. So that is the data format, and then the analysis of analysis logic, that why we are doing this. Along with that, obviously, it is kind of an interactive place. So there is concept mapping, there is question analysis, and then there is functional linkage by using quizzes and understanding that how it binds together. Now, the projects. So here in projects, you will find research examples. So there's a discrimination or differentiation between courses and projects, which are bundled, placed together in the courses tab on Learn OmicsLogic. Once you do the course, you have to start first with the course, and then you have to move to the project. So you do a course on genomics, and then you do a project on SARS-CoV-2 would make sense, because you have learned about the technology. Now you're learning about the application. Similarly, when you are doing transcriptomics, the next thing that you can try to do is maybe a project on modeling, precision medicine, or the mouse PDX model, mouse PDX project, because that talks about a different type of omics data and how it could be integrated, and why and how transcriptomics data is used in such projects. So we will cover all of that in today's session. Nothing to be worried about. We are just going for a quick overview so that you understand that how this entire thing is structured and could make sense for you to do this training with us. So following with that, the most interesting part is the T-Bioinfo platform, which enables us to do research by using a point and click system. So the point and click system is built for you to understand the logic, that what application, how these algorithms work, what are the different parameters these algorithms use, what are the logical flowchart, how one kind of data travels from one algorithm to the another, so that we can get the outcome of this complex analysis. So all of that is being simplified on T-Bioinfo platform, and that's one of the key reasons that we are using T-Bio for our educational trainings and also for research. Now, all of this is appreciable to the research community, because whatever you're gaining is really important in understanding of biomedical or research. So your progress, your badges, your certification, you can leverage, you can showcase to the community, to the world, because this is kind of something that is going to be really helpful if you are going to start applying research. So three things. First, learn omics logic. Second, server T-Bioinfo. Third, code playground. So we learned about OmicsLogic. We have had some idea about server, and then we are going to learn about the code playground as well. So bioinformatics in R and Python. One of the things that we've been talking about to all of you is that how simple and how easy it is. Now, sometimes it requires you to go for certain different challenges because you know that biological data is so complex, and there is always challenges with integration, with understanding that what kind of data, what kind of algorithm is actually suitable for what kind of data, because of the heterogeneity in this data. There is so much of information, and that too is been spread across different type of data types. Now, in to be able to do some of the downstream analysis, and if you're thinking in the context of generating graphs for publication or presentation, doing analysis by using machine learning, dimensionality reduction, or applying deep learning. So those are the things that are some of the applications that we have put together for bioinformatics in R and Python. And we also have a playground where you can actually practice it all on the web, on a portal, sorry, on your browser. You don't need to install R and Python to your local computer and face challenges with library loading and all of that. But you can do that as well, and we will be able to guide you to be able to do that after the workshop during this 1 month period of time. But in this workshop, we are going to use the code playground and the snippets that you can see on the courses. So one of the reasons that we do this, and basically for you to learn visualization of genomic data, to do exploratory analysis and hypothetical testing. So all the omics data, not only genomics, but we can do a lot of exploratory analysis, and we can test our hypothesis when we see patterns in the data, whether the data is following the trend that we are hypothesizing it to or not. I hope that this, whatever I'm discussing so far, is kind of makes sense and clear. If not, please let me know. So again, one of the reasons for to simplify coding is using this code blocks and using the tutorials. So anyone without a background of coding can start applying coding for bioinformatics. So it is not a hardcore coding course. It is a coding for biologists, people who would want to use these technology, use these languages for their own analysis, not just get mastered and start developing applications. If you're interested in developing applications and getting mastered in R and Python, there are like plethora of courses online that would supplement this course for you to to be able to do that.